10.4 Blood and Capillary Exchange. The blood consists of a liquid portion, which is called blood plasma. About 55% of whole blood is plasma. This is the liquid component. And then 45% we call the formed elements. The formed elements includes red blood cells, white blood cells, and platelets. Red blood cells are manufactured in bone marrow. They are a biconcave disc. We'll see a diagram in a moment that illustrates this specific shape. They have no nucleus. The nucleus is ejected as they are maturing in the bone marrow. So essentially they are a bag of hemoglobin. Hemoglobin is the pigment that carries oxygen. These numbers are just astonishing. 4 to 6 million red blood cells per cubic millimeter of blood. So that's just a tiny little drop of blood can have as many as 4 million red blood cells. If you have low numbers of red blood cells, this results in anemia. It can be either low numbers of red blood cells or low quantities of hemoglobin. The lifespan of a red blood cell is about 120 days, and then they become damaged, they're worn out, and they are removed from circulation in the spleen. So here is that shape. It looks kind of like a donut where the hole hasn't been punched all the way through. They are so tiny. You see a diagram here. This also illustrates just how tiny blood capillaries are. If there are 4 million red blood cells in a tiny drop of blood, and capillaries are so small that these red blood cells have to flow through single file. So the shape of the red blood cell is very important because that allows it to be flexible, and so it can navigate through these microscopic capillaries. Something like sickle cell anemia is a mutation that results in a misshapen red blood cell, and it becomes sickle-shaped and rigid and will not flow through capillaries as efficiently. The hemoglobin molecule is inside the red blood cell. This is 4 separate protein chains that form the hemoglobin molecule. There is an iron-containing heme group in each of these protein subunits, and the heme group is where the oxygen actually binds to be transported in the blood. Unfortunately, we occasionally hear about individuals that die from carbon monoxide poisoning. Carbon monoxide will also bind to the hemoglobin, but it won't let go. And so if carbon monoxide is bound up, then the hemoglobin cannot carry oxygen. In addition to the red blood cells, the other formed elements are the white blood cells, or leukocytes. Some of these are called granular leukocytes because whenever we stain them and look at them in the microscope, they appear granular. So the basophils, the eosinophils, and the neutrophils are 3 specific types of leukocytes. They each have a specific function in helping to keep us safe from pathogens. So you can see here, this is a chart from your book that lists these 3 specific types along with their function. 2 other types of leukocytes are grouped together as the agranular leukocytes because they do not contain the granules when stained and viewed under the microscope. Lymphocytes and monocytes are agranular leukocytes. Monocytes are interesting because they can actually leave the blood vessels and migrate out into peripheral tissues, and when they are outside the blood we call them a macrophage, and their job is to phagocyze or gobble up pathogens and cellular debris. We'll see a picture of that in just a moment. The other formed element is the platelet. These are cellular fragments which are responsible for blood clotting. During the blood clotting reaction, these stick together and initiate that process. Here's a diagram of a macrophage, and it is engulfing some bacteria. So, picture taken with a scanning electron microscope, and you can see these cytoplasmic extensions from the macrophage grabbing and engulfing bacteria. Blood plasma is about 90% water. And then there are many other things that can be dissolved in that. A number of different plasma proteins, salts, ions, the gases, oxygen and carbon dioxide, any nutrients, any waste products, any hormones, any vitamins, anything that normally needs to be transported in blood can be found dissolved in blood plasma. Capillary exchange. This is where exchange of all gases and nutrients, waste products, takes place. Recall capillary walls are very thin, a single squamous epithelial cell thick. These are very permeable to water, but they are also permeable to small solute molecules. In the capillary beds, blood pressure is going to force water out of the capillaries, along with some of these small solutes, and osmosis is going to oppose that. So we have two opposing forces. Blood pressure is forcing liquid out, osmosis is trying to pull the water back in. However, overall, there is some water that is left behind. There is more water that is forced out of the capillary bed than is drawn back in. And when this water is left outside of the blood vessels, it is called tissue fluid. It's very important because it bathes other tissues and is a medium for diffusion of material between blood and peripheral cells. So this is a scary looking diagram, but really what it is illustrating is that on the arterial end we have both blood pressure and osmosis opposing one another, but the net pressure on the arterial end is out. If we look over on the right-hand side, this would be the venous end of the capillary bed. We still have blood pressure, although the blood pressure is lower because we're further from the heart. And on the venous end, we have a net pressure in. But if we compare the net pressure out on the arterial end versus the net pressure in on the venous end, there is an excess pressure out, and this is what results in tissue fluid. That is water that has been forced out of the capillary beds and remains as tissue fluid. So in this diagram we see blood vessels, arteries, the capillary bed, and the venule. And so fluid is forced out of the blood capillaries and it can accumulate as tissue fluid, but at some point we have to return that fluid to circulation. And so we have another system which is called the lymphatic system that we will examine in the next chapter, and the lymph system contains some tiny lymph capillaries, and this will pick up that excess tissue fluid and eventually returns it to circulation. Now the lymph vessels are not a circuit. These little lymph capillaries are dead-end vessels, but they have some little trap doors in them. And that is what allows some of the tissue fluid to be forced back in through these trap doors into the lymph capillaries. They are going to merge into some larger vessels and then eventually into the circulatory system.